Scientists can sequence its genome. They can trace every link in its transmission chain. They can prove, with molecular certainty, that it passes from one human body to another. What they cannot do is explain why it is the only one. That is the central question. Not how Andes virus kills, that part is well documented, clinically unremarkable in the taxonomy of hemorrhagic fevers. The question is why, out of every hantavirus on Earth, this single species learned to do something the others never have. And why, after 3 decades of study, no one can explain the mechanism. Here is the rule: not a guideline, not a tendency, not a probabilistic lean, an iron law of virology, confirmed across continents, tested by epidemiologists in dozens of outbreaks, reinforced by molecular surveillance spanning 30 years. Hantaviruses are rodent-borne zoonotic agents transmitted to humans through inhalation of aerosolized excreta, dried urine, feces, saliva, produced by infected rodents. Person-to-person transmission has not been reported despite extensive epidemiologic studies. The CDC wrote those words, the WHO ratified them. Every textbook published between 1993 and 2019 repeated them as settled science. The number of hantavirus species capable of causing human disease sits somewhere between 20 and 30. The number that have demonstrated confirmed human-to-human transmission is 1. Andes virus was first identified in Argentina in 1995, during an outbreak in the Andean region of Patagonia, a landscape of glacial lakes and sparse settlements where the cola largo, a species of long-tailed pygmy rice rat, carries the pathogen in its blood without apparent illness. The virus was named after the mountain range. The mountains, characteristically, offered no explanation. The 1995 cases distinguished themselves not through lethality, though that was considerable. Case fatality rate, 40%, a number that places Andes virus in the same lethal stratum as Ebola during certain outbreaks. What distinguished them was the pattern. Secondary cases appeared in individuals with no documented rodent exposure. They had touched no nests, cleaned no barns, inhaled no dust from abandoned structures. Their only exposure was to other sick humans. This was not supposed to happen. Sin nombre virus, the hantavirus responsible for the 1993 Four Corners outbreak in the American Southwest, shares genetic architecture with Andes virus. Both belong to the same viral family. Both cause hantavirus pulmonary syndrome, the same catastrophic flooding of the lungs. Both kill at comparable rates. Sin nombre has been studied exhaustively since its discovery. In 3 decades of documented outbreaks across North America, it has never once jumped from person to person. Not a single confirmed case. Andes virus does it routinely. The paradox is not medical, it is epistemological. The tools designed to predict viral behavior, phylogenetic analysis, receptor binding studies, genomic comparison, cannot account for this singular exception. By every measurable standard, the two viruses look like close relatives following the same playbook. One of them is not. Dr. Emily Abdoler, an infectious disease specialist at the University of Michigan, stated it with a kind of clinical bluntness that only arrives after the ambiguity has been fully exhausted. Of 20 to 30 hantavirus species worldwide capable of causing human disease, only one, the Andes virus, found in Argentina and Chile, has been implicated in human-to-human transmission. Not a hypothesis offered for peer debate. A summary of settled, reproducible evidence. What followed that confirmation was not a breakthrough. It was a silence. The mechanism remains unknown. Genome sequencing reveals no unique mutations in the Andes virus that would explain its transmissibility. Viral load studies show nothing anomalous. The biology offers no answer, even as the epidemiology keeps generating the same inconvenient data. Since 1995, more than 1,200 cases of hantavirus pulmonary syndrome have been confirmed in Argentina alone. More than 100 cases occur each year across Argentina and Chile. Mortality rate, between 21 and 50%, depending on the outbreak. These are not projections. These are autopsies. The WHO, measured, diplomatic, institutionally allergic to alarm, has acknowledged in its own fact sheets that human-to-human transmission of Andes virus, while uncommon, is documented. The word uncommon is doing significant labor in that sentence. So consider what sits on the table. A virus that kills 4 out of every 10 people it infects. A transmission pathway that violates the foundational rule governing its entire viral family. 30 years of molecular investigation producing no explanatory mechanism, 100 new cases every year in the shadow of the Andes. This is not a pandemic. It is something potentially worse, a proof of concept, evidence that the boundary between rodent-borne and airborne, between animal disease and human contagion, is not as fixed as the models assume. If one hantavirus can cross that line without a detectable genetic reason, what exactly is preventing the others from crossing it next? November 2018. Epuyén, Argentine Patagonia. 3,000 people pressed against a lake, forests thick enough to muffle screams. The kind of town where social gatherings are not optional, where everyone embraces the same friends, shares meals in the same small rooms, breathes air that has already passed through a neighbor's lungs. 3 residents did exactly that before the fever arrived. Muscle pain followed. Then the prodromal whisper, the body's last polite warning before the drowning begins. 34 infections within weeks. 11 dead. The reproductive number reached 2.12 before containment dragged it below 1. For context, seasonal influenza manages 1.2 to 1.4. A hantavirus, by every model constructed over 3 decades of surveillance across dozens of species on multiple continents, should register 0 for human-to-human spread. 0. The entire taxonomic family had earned that number through exhaustive study. Andes virus posted a figure that belonged to measles, not a rodent-borne zoonosis confined to the southern cone. Molecular sequencing eliminated ambiguity. Phylogenetic analysis traced each case with the precision of a genealogist mapping royal bloodlines. Secondary infections carried viral genomes nearly identical to their index contacts. The majority of those infected had no documented rodent exposure. The virus had not leapt independently from animal to human 34 separate times in the same small town. It moved, with methodical indifference, from person to person to person. Not inference, proof. Argentina imposed something Epuyén had never experienced, and the country had not deployed in modern memory for a hantavirus full quarantine. Not advisory isolation. Not recommended distancing. The government restricted movement in and out of the town, established epidemiological cordons, deployed contact tracing teams to reconstruct every handshake, every shared cup, every moment of proximity that might constitute transmission. The quarantine held. By early 2019, the chain was broken. Quarantine answers containment. It does not answer causation. Genome sequencing of the Epuyén strains revealed no novel mutations, no insertion, deletion, or recombination event that would confer transmissibility. Surface glycoproteins, the machinery by which a hantavirus attaches to human endothelial cells, showed no structural divergence from non-transmissible strains. Viral load analyses in hospitalized patients fell within ranges observed for sin nombre virus in North America, a pathogen that has never once passed between humans despite thousands of confirmed cases. Every molecular assay designed to identify the mechanism of person-to-person spread returned the same verdict: indistinguishable. The virus doing something unprecedented looked, under every available instrument, identical to viruses that could not do it at all. Epidemiology confirmed transmission. Virology could not explain it. 2 disciplines designed to operate in concert, each validating the other, produced conclusions that did not reconcile. One said the event was real. The other said the event was impossible by its own framework. The virus displayed no such confusion. It crossed into Chile. A subsequent case, linked by travel history and confirmed by molecular analysis, demonstrated that Andes virus respected neither the rules of hantavirus transmission nor the sovereignty of international boundaries. Chilean health authorities initiated their own surveillance protocols. The Western Hemisphere's only person-to-person hantavirus had become, in the most literal sense, a transnational concern. The Apuyen outbreak ended. The accumulation did not. Since 1995, over 1,200 confirmed cases of Andes virus hantavirus pulmonary syndrome have been documented in Argentina alone. Case fatality rates oscillate between 21 and 50% depending on the outbreak, the region, the hospital infrastructure available. The WHO acknowledges human-to-human transmission as documented but uncommon. The CDC notes, with a careful phrasing of an institution aware of what it does not know, that person-to-person transmission has not been reported for any other hantavirus despite extensive epidemiologic studies. Uncommon, documented, unexplained. Three words that, in virology, should never describe the same pathogen for 3 consecutive decades. Yet here they sit, undisturbed, attached to a virus that has been sequenced, quarantined, modeled, published, and studied by laboratories on 3 continents. No consensus on the single question that matters. The outbreaks have been contained. The dead have been counted. The genomes have been filed in databases that will outlive everyone who contributed to them. And the question, what makes Andes virus capable of something no other hantavirus can do, remained, after 30 years of investigation, exactly where it started. Then researchers stopped looking at the virus. They began looking at the space between the virus and... The question is not whether Andes virus transmits person to person. That debate ended in Apuyen. The question is whether the exception is a preview. Researchers did what researchers do when confronted with the inexplicable. They sequenced everything. Every nucleotide of Andes virus was mapped, cataloged, and compared against its closest relative, Sin Nombre virus, the North American hantavirus responsible for the 1993 Four Corners outbreak that killed half the people it infected. The comparison should have revealed the mechanism: a mutation in the glycoprotein, an alteration in receptor binding, some molecular signature that would explain why one virus leaps between humans while the other does not. They found nothing. The genomes are nearly identical in the regions that matter. The surface proteins that determine how a virus enters a cell, the replication machinery that determines how it spreads, functionally indistinguishable. If you showed a virologist both sequences without labels, they could not tell you which one transmits between people and which one does not. The difference, if it exists at all, is written in a language we do not yet speak. This is not a gap in knowledge. It is an admission of ignorance dressed in technical language. The Syrian hamster does not care about our ignorance. When researchers inoculated hamsters with Andes virus and placed them in cages with uninfected animals, transmission occurred. Reliably. The virus moved from host to host without rodent excreta, without environmental contamination, without any of the mechanisms that define hantavirus ecology. It simply moved. The hamster model confirmed what the epidemiologists had suspected. Andes virus behaves differently not because of what it is, but because of what it does. The distinction matters. A virus defined by its genome is a static thing, a sequence to be decoded. A virus defined by its behavior is a process, and processes evolve. Sin Nombre virus does not transmit between hamsters. Neither do the dozen other hantaviruses tested in the same experimental system. Only Andes. The exception remains exceptional, and exceptions in virology have a habit of becoming rules when the conditions are right. Here is what we know about person-to-person transmission of Andes virus. It requires close contact, prolonged exposure, and, this is the detail that should concern you, respiratory involvement during the symptomatic phase. The virus does not spread during the incubation period, when patients feel well enough to move through the world unnoticed. It spreads when they are sick enough to be coughing, sick enough to be struggling for breath, sick enough to be producing the kind of respiratory secretions that carry viral particles into the air. This is not efficient transmission. It is not measles, which can infect you from across a room. It is not influenza, which spreads before you know you are ill. But it is transmission, and transmission is a threshold. Once crossed, the question is no longer whether a virus can spread between humans, but how easily. The case fatality rate is 40%. The incubation period is 2 to 3 weeks. The transmission window is narrow, constrained to the days when patients are symptomatic enough to shed virus but not yet dead or hospitalized. These are not the characteristics of a pandemic pathogen. They are the characteristics of a pathogen that is not yet optimized. In 1996, a physician in Bariloche, Argentina, treated a patient with Hantavirus pulmonary syndrome. 3 weeks later, the physician developed the same disease. The epidemiological investigation found no environmental exposure, no contact with rodents, no plausible source of infection except the patient. The physician survived. The patient did not. The case was documented, filed, and cited in every subsequent paper on Andes virus transmission. It is treated as evidence of human-to-human spread, which it is. But it is also evidence of something else. Healthcare workers are not dying in large numbers. If Andes virus transmitted easily in clinical settings, we would see clusters among medical staff. We do not. The Bariloche case remains notable precisely because it is rare. This should be reassuring. It is not. Viruses do not remain static. They adapt, they mutate, they explore the fitness landscape available to them. Andes virus has crossed the threshold into human-to-human transmission, but it has not yet optimized for it. The question is not whether it will. The question is whether we will notice when it does. The scientific community continues to monitor. Outbreaks are contained. The virus has not evolved further yet. This is not reassurance. This is the concession that some threats remain opaque even under full scrutiny, that the machinery of modern virology can surround a pathogen completely and still fail to penetrate its central logic. Andes virus remains confined to South America, predominantly Argentina and Chile, where more than 100 cases of Hantavirus pulmonary syndrome surface each year. Surveillance systems function. Quarantine protocols, as Epuyen demonstrated, can reduce the reproductive number below 1. The case fatality rate persists between 21 and 50% depending on the outbreak, but the absolute death toll stays modest by global epidemiological standards. By every metric designed to measure danger, the virus registers as manageable. Managed, however, is not the same as understood. The distinction matters more than any case count. Genome sequencing has mapped Andes virus to the nucleotide level. Epidemiological modeling has traced its transmission chains across households, social gatherings, and at least one hospital ward. Viral load analysis has measured its replication kinetics in human tissue. Every instrument available to 21st century virology has been aimed at a single question: why does this hantavirus, and no other, pass between human bodies? The instruments returned clean data, reproducible results, and no answer. Not an incomplete answer. Not a provisional hypothesis awaiting confirmation. Nothing. This is the paradox that should unsettle anyone who assumes science progresses linearly toward resolution. 3 decades of investigation have produced over 1,200 confirmed cases in Argentina alone. Molecular proof of person-to-person transmission chains, who acknowledgement of the phenomenon, and avoid precisely where the explanatory mechanism should be. Consider what the absence reveals at the structural level. Virology's predictive framework operates on a tacit assumption that transmissibility correlates with identifiable molecular features. Receptor binding, affinity, polymerase fidelity, glycoprotein conformation, these are the variables the models interrogate when assessing pandemic potential. The framework does not merely fail to explain the exception, it fails to detect the exception as exceptional at all. What this exposes is not a gap in knowledge about one obscure Patagonian pathogen. It is a lacuna in the epistemological architecture governing pandemic preparedness worldwide. Risk assessment models evaluate known variables. Andes virus demonstrates that the variable responsible for the most consequential shift in hantavirus behavior, the leap from zoonotic to interpersonal transmission, is not among them. Sin nombre, Pumala, Sol virus, all classified as rodent-only transmission based on epidemiological studies conducted primarily during outbreaks, in settings where contact tracing is incomplete and molecular sequencing is not always available. The WHO's own language is precise. Human-to-human transmission remains uncommon. Uncommon is not impossible. Absence of evidence, as any first-year epidemiologist recites, is not evidence of absence. Most hantavirus cases occur in rural, under-surveilled regions where the infrastructure to distinguish a rodent exposure event from a close contact transmission event simply does not exist. No vaccine protects against Andes virus. No antiviral treatment has demonstrated efficacy in clinical trials. Supportive care, mechanical ventilation, hemodynamic management reduces mortality from roughly 50% to 30%. That constitutes the entire medical arsenal directed at a pathogen that has already crossed the threshold every other hantavirus has respected. The central question has a direct answer, and it is this. The mechanism is unknown because the existing framework lacks the conceptual vocabulary to identify it. Andes virus did not outsmart virology. It revealed that virology's predictive models contain an unexamined assumption that transmissibility must leave a molecular fingerprint detectable by current instruments. The exception persists not because science has failed to look, but because science has been looking with tools calibrated to detect a signal that may not exist in the form expected. The significance extends beyond a single virus circulating through Patagonian towns. Andes virus establishes a principle with implications for every zoonotic pathogen under surveillance. A virus can acquire the capacity for sustained human-to-human transmission without triggering any of the molecular alarms designed to detect precisely that shift. The models that govern global pandemic preparedness are built on observable precedent. Andes virus is observable precedent that the models are incomplete, and it has been sitting in plain sight, fully sequenced, and thoroughly documented for 3 decades. Every observation has a sample size, and every rule in virology is only an observation that has not yet encountered its exception.